because then I can actually say that I know no chemistry and get away with it. If I say that in front of a chemistry crowd,
I see few chemists, but there are only a few.
They're not going to get angry when I say I got a Nobel Prize in chemistry, but I know no chemistry. Okay. So, oops, I don't have.
Oh yes, microphone. You have to do something with the microphone.
Oh, I just need to flip a switch on.
Microphone? Pardon?
How's that?
Le micro, j'ai oublié le micro. C'est bon, it's working now.
It's working now? Okay, all right. So, so a large part of my career has been with working on the problem that if that's the size of a protein molecule, then when you look at it in a regular optical microscope, what you see is that big fuzzy blob. And so on the one hand, this is bad news because if we want to understand how inanimate molecules can assemble to create an animate cell, it's too coarse by about a factor of 100. But on the other hand, it's good news because that fuzzy blob is still 100 times smaller than the cell itself. So that, of course, for hundreds of years, people have learned a lot of cell biology by applying the conventional optical microscope. So in the first half of my talk, I'm going to tell you about ways to get beyond that diffraction limit. And then I'm going to turn around and talk out of the other side of my mouth and say, hey, wait a minute. There's a lot that we as physicists can contribute to make microscopes better even at the diffraction limit. So I had several forays in trying to do super resolution. The one that finally worked reasonably well was that if the problem is that the molecules in the sample are so close together that the fuzzy blobs created by each molecule overlap, what if the light from each molecule was somehow different? For example, if all those molecules glowed in different colors. Then if we had a spectrometer on there, we could isolate them in a higher dimensional space. But once the molecules were isolated from one another, it's relatively easy to fit a function to that big fuzzy blob and find the center of that function to much better precision than the diameter of the fuzzy blob. And the more photons you get, the higher the precision of your estimate of where the center is. And so it's fairly easy with a few hundred photons to get sub 10 nanometer precision. And then if we could do that with every molecule, if they were sufficiently isolated, we would just have a molecular resolution map of the sample. So that was an idea I pitched after I left Bell. I got that idea while I was pushing my daughter in a stroller while I was unemployed, and then I just published that on my own. But there was a catch with this idea, which is that in a normal cell, there might be hundreds or thousands of proteins in a diffraction-limited spot. So you need really good discrimination of individual molecules, see one on top of 1,000 others. And that didn't really exist at that time. So it was just a sort of Gedanken experiment. So fast forward 10 years after I'd failed at my dad's company and was looking for something new to do, when my buddy Harold Hess, who had been at Bell with me, and I learned about a new type of fluorescent protein, which is initially non-fluorescent unless you shine violet light on it, in which case it becomes activated, and then if you shine blue light on it, it would glow green. And so we immediately realized, this was developed by Jennifer Lippincott-Schwartz and George Patterson at NIH, and we immediately realized when we heard about this that this was the missing link to make that idea that I had pitched a decade before to work. Because you could transfect your cell with this, get this photoactivated protein labeled on any protein you cared about, turn on the violet light so low that only statistically a few molecules are going to come on across your sample. They make these fuzzy balls, but because they're separate, you can find their centers. Those either bleach and turn off, or they turn off by other means. And then you can find those centers and do that again, turn on the activation, find the centers, turn them off, on and on again, with different subsets until you start to build up this super resolution image. So it's the idea I had a decade before, but instead of wavelength, doing it in time and turning on the molecules. So that turned out to work very effectively and quickly, and so we went basically from the idea of Palm to having all the idea in the science paper that got me the Nobel within less than 6 months. And so this is an example. The lights in here are really bright and the monitor is dim, so it's hard to see, but this is the diffraction limited image, this is the Palm image. for photoactivated localization microscope. These are lysosomes in a cell, and we're doing a thin cut section to look through those lysosomes. If you zoom in here, you can see the kind of detail you can get. So if everything goes right, and you can take this technique down to about 20 nanometers, everything going right means that you have to densely label the sample, and I'll get into that. So Harold and I, at the same time, both got jobs at the Genelia Research Campus of the Howard Hughes Medical Institute, which was modeled after Bell Labs. And so once we were there, we started to do applications of Palm. Here we were able to look at chemotaxis receptors in E. coli and show that there's a wide spectrum of cluster sizes. That spectrum is actually predictable in terms of a stochastic model of self-assembly, where the protein monomers are inserted into the membrane randomly, diffuse randomly until they stick, and the probability of sticking is just proportional to the existing size of a cluster. Here we looked at focal adhesions, which are the points of contact of the cell to the extracellular matrix, and so kind of the feet of the cell. And there's about 90 different proteins in these focal adhesions, but in this case we were looking at a pair, and vinculin and paxillin, and at the diffraction limit they look like they're co-localized, but with higher resolution they form little aggregates that are spatially segregated. In another example with Bob Teach and Genelia, we were able to show that these green dots, which represent core promoters which are necessary for transcription of DNA, are spatially segregated from the genes which hug up against this red nuclear membrane. And that spatial segregation is one means by which the cell uses to regulate when certain genes are turned on. And in another example, we were able to look at cultured neurons and show in the spines on the dendrites of these neurons that the cytoskeleton, which gives these things their shape, are only polymerized at certain discrete places in these dendritic spines. So as I mentioned before, one of the major problems of any form of superresolution, not just this Palm technique, is that you have to have a very dense number of fluorescent labels. So if I had like the sine wave here and I sample it only every half period, I could miss that pattern altogether. So that's what's known as the Nyquist criterion. If you want resolution X, you have to sample at least every X over 2. So if I want 20 nanometer resolution in a diffraction limited spot, that would mean I'd have 500 fluorescent molecules. And I need to be able to see one on top of that 500. That's doable with the on-off contrast ratio of fluorescent proteins. But what's hard is to get 500 in a diffraction limited spot. Because normally, when you transfect cells or otherwise label cells at the diffraction limit, you're working at much lower levels of labeling. So a lot of the onus to get these super resolution techniques to work well isn't so much on the instrument developers anymore, it's on the biologists who do the sample preparation. And it's really difficult to do that properly. So this is almost hopeless to see in this slide, but it's even harder to notice that here's an electron micrograph in which we blurred to about 20 nanometer resolution. If instead we start popping molecules onto the structure in another Gedanken experiment, if we do that at that Nyquist criterion, there's still so much stochasticity in where the labels fall that it's not a good enough criterion. You really have to go almost 10 times beyond the Nyquist criterion of labeling to really decorate the structure with high fidelity. And so that means it's not 500, it's 5,000. And it gets really, really difficult. It's doable, but it's really difficult to do that. But that's true of most of the methods of super resolution, where what you do is you load up your fluorescence in your cell, either in a fixed cell or a live cell you then fix, and then you image it. And when you do that, you basically have n number of molecules to begin with, and that's it. And that, however many you got, that's your budget. But there's another method of super resolution that came out in the same year as our Palm paper, which works differently. So in this method, it's called paint. The molecules are actually in the cell media, not in the cell itself initially. And normally they're whizzing around so fast that under their Brownian motion you can't see them at the capture rate of the cameras. But when they stick and bind to their targets, then they're fixed and they're a little glowing star, and then you can localize those. So that's been used to great effect to look at, for example, DNA origami structures with DNA oligos that transiently bind to complementary pairs that are within these oligos, or within these origami structures. But it's difficult to do this normally in high density in whole cells. And the problem is that the media itself is fluorescent, and it creates so much background that you can't see single molecules on top of all of the other out of focus molecules that are also glowing. So that's where we brought to play one of the technologies I'll go into more detail later, which we call lattice light sheet microscopy. So normally when you do single molecule experiments, you illuminate the entire sample with wide field light. So here you can see a spheroid of about 35 microns big of embryonic stem cells. And if you illuminate the whole thing to try to look at these transcription factor molecules in there, all the out of focus molecules make it impossible. It's just a haze. But if you take a light sheet that we can create that's thinner than the depth of focus of our detection objective to look at the molecules, then if you park that at one plane, then only molecules that are in focus get illuminated. And then you get very high signal to noise to be able to see those molecules. And in this case, in a couple studies, we use this to study the transient kinetics of the binding of transcription factors, both specific and nonspecific, to DNA. Now what we can do, therefore, is we can use our lattice light sheet as a way to get around the problem in paint of the whole media glowing, and then be able to look at one plane at a time, do our paint there, go to the next plane, do it again, go through the whole volume of the cell, then come back to the beginning, because then new molecules will have bound then to the thing out of the solution. You do it again and again, and you do that thousands, hundreds of thousands of times. And instead of in a normal palm image where you might have localized a few million molecules, now you're talking about localizing 500 million molecules. So now your images start to look like 3D EM images with the amount of information that they contain. And so here we're doing some line cuts through these, and you can see that by this metric that you can get down to 70 nanometer resolution fairly quickly by doing that. Fairly quickly, not in time, but in simplicity. This is another example where we're the same example where we're actually characterizing the resolution not by line cuts, which I believe is a bad metric, but rather by a combination of the localization precision and the density of localization events that we're able to see. So another major advantage of this approach is that there's been a lot of people in the super resolution field who've been trying to do correlative electron microscopy with super resolution microscopy, because the EM gives you global contrast of all the membranes, and then the super resolution gives you protein specific contrast. But it's extremely difficult to do because the heavy metals which are necessary for EM contrast are lethal to fluorescence. And so you're always trading off between the amount of EM label you can put in versus the amount of fluorescent label that you kill. And I already said you need a lot of fluorescent label for this to work well. But with the paint approach, we have a paint probe which can also stain all the membranes to give us global contrast. And we can then do that in the exact same instrument in which we do the palm. So in this case, we're doing 3-color imaging of all the nuclear membranes. We're looking with another probe at the DNA, and then a third is looking at, by protein contrast, looking at the nuclear envelope, and then we can see all three. So the other advantage of paint is that we can extend this from thin samples, which is usually what super resolution is limited to much thicker specimens. So in this case, we're looking at a whole mitotic cell in cytokinesis. Here are the retraction fibers. Here are these little fibrils that form when the membrane has to stick out as it reduces its volume, but to maintain surface area. And so this is 2-color imaging here. We can compare that to the other modes of this light sheet microscope I'll talk about earlier, and there's the clear resolution improvement. by going to localization microscopy. So this is a different example of looking at mitotic cells, a pair of cells, one in prophase, one in metaphase. And in this case, what we're doing is comparing to another technique I'm going to talk about in a few minutes called structured illumination microscopy. So that's another method to get at least twice beyond the diffraction limit. It's often used in 3 dimensions, but it's really limited because it's a wide field technique and there's always a lot of background. And so if you go into something this thick, you get nothing but artifacts when you try to look at the sample. But again, because of that property, the lattice light sheet, that we restrict the excitation and have the almost zero background from the out of focus, the paint is able to maintain its resolution even in these thick specimens. So the largest thing we've looked at so far by the lattice paint is a whole zebrafish. Not that we've looked at the whole fish, because that's huge, but we've looked at the neuromasked organ on the zebrafish along its lateral line. So in this case, we've been able to look about 30 microns deep. In this case, it's about 1 billion localization events. So one thing paint is not is fast. So to localize 1 billion molecules took 10 continuous days of imaging in the microscope. And yet we still can register every molecule relative to one another over those 10 days to precision of better than 10 nanometers. So one of the limits of the paint approach is that as you go deeper, there's a chance that the molecules, before they bind on their way down to getting deep, that they can bleach because the light sheet keeps coming through and through and through, and the deeper you go, the more times it gets whacked by the light sheet before you get there. So eventually you run out of the ability to go deep, but it depends upon the molecular weight of the paint probe. A smaller molecular weight one can diffuse faster and hence go deeper. So all of that is fine, but everything I've showed so far is on a fixed dead sample, okay? And about 98% of all of the results of super resolution microscopy have been on fixed dead samples. There's a lot of problems with that, among them that fixatives are known to affect the ultrastructure by their very nature, and so you're not really looking at something physiological right to begin with. But the real problem is that the thing that defines life is that it's animate. And every living thing is a pocket of reduced entropy through which matter and energy is flowing continuously. So while structural imaging will always be informative, the only way we're really going to understand how those inanimate molecules can make an animate cell is by looking across all 4 dimensions of space-time at the same time. Okay? So that's going to be our ultimate goal. But the first step is we need to look at live things, right? That's the message. What are the challenges of doing that? Well, we can go right back to that Nyquist criterion I mentioned earlier. If I want to increase the resolution n times, and I want to do it in d different dimensions, then if my voxels have to be n times smaller in each dimension, so I'm going to have n to the d times as many voxels in my higher resolution image. If I want to have the same signal to noise I had at the diffraction limit, each one of those voxels has to have the same number of photons. So that means the overall image has to be composed of n to the d times as many photons as before. What that means is that based on various claims in the literature for live imaging, you're asking the cell in one frame to give up anywhere from 4 times to 14,000 times as much light to create your image as in a diffraction limited image. If any of you do any kind of live cell fluorescence imaging, you'll know there's no cell out there that you can image for even 1,000 times without it being completely bleached. And so most of these metrics down in this range are nothing but fantasy. There's no way you can get to those levels. And furthermore, this is just to get to one frame. If you're going to do live imaging, it's composed of multiple frames of data, right? And so you have to parse this photon budget across those multiple frames. So it gets really hard. But it's also hard not in terms of just photons, but also in terms of intensity. Because assume I wanted to get the data at the same rate that I got at the diffraction limit. Then my n to the d more photons means I need a rate of photon emission n to the d higher, which means my photons going in have to be n to the d higher, which means n to the d higher intensity, which is going to start cooking my cell by having too much light on it. But furthermore, it's worse than that, because if I want to look at a feature that's traveling at 1 micron per second, if I had 1 micron resolution, I can afford to take a second to get the image. But if I want 10th micron resolution, I can only have a 10th of a second to snap that image. So there's a bad relationship that as the spatial resolution improves, the temporal resolution has to move in direct proportion, or else you're going to get motion artifacts. So that means the signal rate now has to be n to the d+1 times higher. Now there's a fork in the road depending on which method of super resolution you want to use. There's one class, which is like structured illumination and localization, which is parallel because it's wide field. And then there's point scanning methods, such as the STED that Stefan Hell got the Nobel for, and his related resolve technique, which started out at least as point scanning methods. The problem with the point scanning method is that only one voxel is contributing photons at a time, unlike the other where they're all in parallel. And so that voxel has to contribute then n to the d higher rate in itself for a total of n to the 2d plus 1. So even something as modest as a factor of 2 improvement in resolution in 3 dimensions would therefore require 128 times the rate of photons coming out, which means 128 times as much light going in. And there are claims of doing live imaging in 3D at 50 nanometers. So that would be then 4 to the 7th. I don't know which would be what, 128 times 128? We're talking, 25,000 times as much light that you would have to get out. It's just not plausible in order to do that. So the moral of the story is that really, although many people in the super resolution field have claimed for years that you can have it all, you can't. And there's always trade-offs. And the higher spatial resolution you want, that means you got more pixels, which is going to take more time to get that data, and it means throwing more potentially damaging radiation at the specimen. So the one guy who appreciated those trade-offs before the rest of us was Mats Gustafsson, who was one of the pioneers of a different super resolution method called structured illumination microscopy. In this technique, it's a wide field method, not single molecule, in which you, instead of illuminating your sample uniformly, you illuminate it with a grading pattern, usually a standing wave that's created right at the edges of the diffraction limit. And then that standing wave beats against the different features inside of the sample to create low-frequency moire fringes, which are within the resolution limit of the microscope and hence detectable. By doing that, and that sort of demodulation approach, you're able to extend the resolution by a factor of 2, because you can't write a finer grading than the diffraction limit itself. So that's one of the reasons the committee cited in not having SIM be part of that Nobel. But I actually think that's a mistake, because I think SIM will actually be the super resolution method that will end up contributing the most to biology. And that's because this limitation of a factor of 2 is in some ways a strength, because A, you don't need to densely label the sample to the same degree as the other methods. B, it requires orders of magnitude less light. Life evolved at 1/10 of a watt per square centimeter. Localization microscopy requires a kilowatt per square centimeter. STED requires hundreds of megawatts to a gigawatt per square centimeter. So they're really not good tools for doing live cell imaging. SIM can get away with 1 to 10 watts per square centimeter, so it's much more in the ballpark of what life can tolerate. But it's also very fast, so it's easy to do sub-second acquisitions. So this is looking at the endoplasmic reticulum inside of a cell. There is no other super resolution method that would get within 1 order of magnitude of being able to look at that same cell for 1,800 time points and do it at sub-second rates. But furthermore, you can push the speed. In this case, it's a zoom in of the ER showing for 600 time points at 50 frames a second what's going on. What you learn after a while in developing these techniques is the cell is a crazily dynamic place, and we really need these dynamic tools to be able to understand how all of this goes on. Oh, I'll skip that one anyway. And this one stopped again for some. So Another thing that you can do is run these SIM microscopes in not a total internal reflection configuration, but close to it. So you can look at thicker specimens and still not illuminate the depth of the specimen, so you have high enough SNR. It's probably a little hard to see on this screen, but the green represent the microtubules inside of the cell. These little bags are lysosomes. And you can watch these lysosomes as they track along and then switch tracks, like switching train tracks and going on other microtubules. Sometimes they stop and sometimes they move like a bat out of hell when there's molecular motors there to really pull them along. But the method that's probably been most used by our collaborators with SIM is to do it in turf, where you use an evanescent field to only illuminate the bottom of the cell. Not only does this reduce the excitation and the damage to the rest of the specimen, it creates very high SNR for SIM and makes it easy to do. So we've used that to study how myosin 2, a motor protein, ends up bundling together in these bipolar filaments and how different types of myosin 2s come together. This is looking at a T cell, such as will fight infection in your body, but it's plopped down on a cover slip that has antigens that provoke what's known as the immunological response, as if it were coming up against a target cell that it was going to try to kill. And then you can see this beautiful symmetry of the actin flow, the cytoskeleton, inside when that synapse is established. So if the criticism of SIM has been that it's only limited to 100 nanometers or a factor of 2 in resolution, what can we do to try to improve upon that? The simple thing we can do is just increase the numerical aperture of our lens. So Olympus sells a lens at 1.7 NA that we can use in turf, which extends the resolution in the green channel from 100 nanometers to 80 nanometers. So we're using that here to, again, I told you about focal adhesions before. Now you're seeing an example where we're looking at the focal adhesions live and in 2 colors by being able to do the SIM. In this case, you're looking at the myosin 2 again, whose role is to bundle actin filaments together and create these actin-myosin forces to drive the motion of the cell. And you can see at the trailing edge of the cell how these forces are predominant. So we can compare what we can get now with these high NA version of structured illumination to the supposedly higher resolution methods of the other super resolution methods. So the first comparison I'll make is to my own work back in 2008 when we were able to demonstrate live cell palm imaging for the first time. In that case, again, we looked at focal adhesions, reported about 60 nanometer resolution as you see here. But the 81 nanometer resolution we have here isn't all that different, yet we were able to take 3 times as many images with the SIM, with 15 times faster, and with 20 times less intensity on the cell. So comparable resolution and a lot better metrics in terms of live imaging. So we've used that to study what's known as clathrin-mediated endocytosis. So the plasma membrane of the cell is the gateway of the cell, and obviously cargoes need to get into that cell. One means is these molecular cargoes come to the membrane, dock, and then there's an invagination of the membrane controlled by this protein called clathrin, which forms this basket that then pinches off the membrane. So in the turf that we're doing here, the evanescent field only lights up this part of the pit, so it ends up looking like a ring when we're able to resolve it with the turf SIM. But now that we can do that, we can actually follow the entire evolution of these clathrin-coated pits from their initiation, their maturation, and their eventually pinch-off and internalization. And so therefore we can then track the lifetimes of these guys, as the different colors here, and then correlate the changes in size to the lifetimes to find the rates of growth and find other correlations as well. So one of the things that we learned is that these pits are formed anywhere, but actually form somewhat randomly, but in certain places there are hot spots. So you can see here this movie starts as a point here, and it's like a kid blowing bubbles, that there's multiple clathrin-coated pits coming out of one site. So something is causing some nucleation there that forms multiple pits at that point. So another thing that's nice about SIM as opposed to the other super resolution methods is STAT, Resolve, Palm, and Storm all require exotic switchable labels or something that can take the gigawatts per square centimeter without immediately going dark. Whereas with SIM, you can use any off-the-shelf fluorescent label, so it's easy to do multicolor imaging. So here's an example where we're looking at a combination of clathrin and actin. So there's been a lot of debate in the literature about the role of the actin cytoskeleton in clathrin-mediated endocytosis and whether it facilitates that. What we found is that in only about half of the cases, right before endocytosis, actin is recruited. But when it is recruited, there's a small but statistically significant shortening of the lifetime. We also find that there are not just individual pits, but these larger aggregates of pits called plaques, which have been reported in the literature before. But what we see is that these plaques are always just aggregations of pits, and they spin off individual clathrin-coated pits from them. We also found, to our surprise, that there were actually tiny rings of actin, like the rings of clathrin. And we thought initially that these would be co-localized and being used to help internalize the pits with their forces. But it turns out that almost none of them are co-localized. And so, you know, there's not a lot of description of these small actin nanoscale rings in the EM literature. And so we're really not quite sure what they are or what their role is. So we described how to use high NA to get to near 80 nanometer resolution, but what if we wanted even higher resolution? Then we need to resort to these photo switching tricks, which are the basis of Palm and STED and Resolved. And so, similar to what's known as the Resolve technique that Stefan Hell developed, one way to do it is to take a photo switchable fluorescent protein, and you first turn your violet light on to activate all of those molecules, and then you come in with a standing wave, which can deactivate all of the molecules except the few that reside. Here's turning them on, and now we're going to turn on the standing wave to deplete all of the molecules except the ones at the nodes of the standing wave. So you have little narrow spikes of fluorescent molecules. Those have higher Fourier components, which allow you to demodulate even higher spatial frequencies and get well beyond the factor of 2 beyond the diffraction limit. So Motz's grad student Hesper published this paper in 2012, where she was able to get to about 50 nanometer resolution by SIM on actin doing this, but it took 900 seconds to get an image. So we tried to think about ways to be able to push this to being able to do live cell imaging, but still to keep the speed up and the intensities low to make it much more compatible for live imaging than the other methods. And so what we realized as we thought about that is that this principle of saturated depletion, which is the principle behind the STED microscope and the Resolve microscope in that previous form I just showed in the last slide, is really not a good way to do live cell imaging, because you're taking all of the molecules up to an activated state, and then the vast majority just go right back down to the ground state. When they do that, they're not contributing anything to the image at all, but every molecule can only undergo a finite number of switching cycles before it's going to bleach. And so you really lose your molecular budget quickly by doing that. Furthermore, it takes a lot of energy to bring the molecules up and down. And second, it takes a lot of time. And time and energy are the enemies of live cell imaging. So what we developed instead was the idea of, instead of activating all the molecules uniformly, is turn on a standing wave of the violet light, which activates a standing wave pattern of fluorescent molecules in the sample, and only a subset of them. And then we turn on a blue light, which reads out the molecules and deactivates them at the same time. And by doing that, we have 2 harmonics instead of 1, and are able to extend the resolution to 60 nanometers instead of 100 nanometers. And so as this movie's been playing back and forth, you can see as we go from diffraction limited to linear sim and nonlinear sim, the gains in resolution as we do that. So once we do that, we can take images as fast as 1/3 of a second. This is a 1/2 a second here. and take anywhere from 1 to 3 or 4 dozen images. It's not as many images as we get with the high NA sim, because this requires 25 images to reconstruct. The high NA sim requires only 9, but it still can do live imaging. It's just, again, the trade-offs: spatial resolution for temporal resolution and number of frames. So we can compare that to even localization microscopy, which is pretty well accepted as the highest resolution method of doing super resolution imaging. This is an example of looking at the actin cytoskeleton in a fixed cell by localization at a reported resolution of 20 nanometers. But whether you look at this image in real space or reciprocal space, it's really comparable to what we get in a live cell with the nonlinear sim, even though our resolution claim is 3 times worse. But furthermore, on a live cell, it's pretty hopeless to get high resolution because you just can't get enough localization events. But if we compare the live cell image here to what we get here and here, we get 34 time points versus 1. We get the data 600 times faster, and we put 20 times less light on the specimen. So if the reason the Nobel Committee didn't give the Nobel in part to SIM was because it didn't have resolution good enough to really be in the same ballpark as STED, Resolved, and localization, this is to prove them wrong, and that's not the case. And that SIM can be every bit as good in resolution and vastly superior, orders of magnitude superior, in terms of intensity and speed of acquisition. So another example here is another method of endocytosis, bringing cargoes in the cell, through little pits called caveolae. And so this is showing the resolution improvement from diffraction limit through linear sim and nonlinear sim. The caveolae are so small that most of them are barely resolved or not resolved even with linear sim, but become resolved with the nonlinear sim. And what we find is in the EM literature, there's a very stereotypical size. 70 nanometers is the size of a caveolae pit. What we find is that there's a great variety of sizes, and we don't know what is the reason for that discrepancy. Some of this could be that we're looking at multiple pits stocked together, but the other possibility is that when you look at an EM image, it's just black and white. You don't know what protein is where. The advantage of fluorescence is you have that protein-specific contrast, and we know where all the caveolin is. So one final comparison then is we can compare that then to the resolve technique I've been mentioning that Stefan Hell developed, because he had a paper last year where he looked at caveolae. So here's the image of caveolae there. Here's the diffraction limited image that we get. Here's the linear sim, which is clearly better already, even at the 100 nanometer level. And then here's what we get with the nonlinear sim. So the moral of the story is, in my opinion, many of the claims in the super resolution literature have vastly overstated their real resolution. And it's because they use these line width metrics, which are really not reflective of what happens in dense samples across most of the cell. And so, you really have to look the gift horse in the mouth if you're going to use these tools, and think carefully about the nature of your specimen, whether it's really suitable to get the level of resolution that they claim. So we have only one good photo-switchable protein for this nonlinear sim, but we can combine it with the linear sim to do multicolor imaging. So this is an example where we're looking at the actin cytoskeleton again, and also RAB5A, which is a marker of early endosomes. So those clathrin-coated pits, once they pinch off and go in the cell, they mature into what's known as an endosome, the next step in the endocytic pathway inside of cells. And so these green blobs are those guys. You can clearly see that at this point, these endosomes don't have a stereotypical round shape like the clathrin-coated pits, but have various strange, weird-looking shapes. I picked out 3 particular examples here that I liked, and I liked them because they matched some EM pictures from a 20-year-old paper. So we can start to clearly resolve some of the shape of these endosomes. We can show that there's dark voids inside, which are either vacuoles or cargoes, and we find all kinds of different dynamics of these guys, either undirected motion, almost like Brownian. Sometimes they're constrained by the cytoskeleton, and sometimes they latch on to probably microtubules or something, and then whoop, they move like a bat out of hell under active transport. So the final example of that two-color imaging is here, where we're looking at actin again, but now in conjunction with alpha-actinin, which is a protein which bundles single actin filaments together into larger stress fiber cables. And so you can see that where the cell needs these larger, stronger cables because it's trying to force out filopodia or force out a membrane ruffle, that's where the alpha-actinin is enriched. We also find, again, our old friends, the actin rings, but these actin rings are always co-localized with alpha-actinin. So they may have some role in bundling them or creating the curvature of these rings. So the moral of the story was, if Motz, by backing off from the direction that everybody else in the field was going and saying, I'm happy with 100 nanometer resolution because I can do all of these other live cell things, he had a whole playground to himself in order to do that. So it begs the question, what if we back off even further? What if we go all the way to the diffraction limit? Is there anything we can do to improve live cell imaging in 3D that would be as much of an improvement in terms of the speed and the low damage that we do to cells in imaging them in 3D as Palm was in terms of the spatial resolution? And so the good news is that there's a lot of opportunities there, because the most common tools for live 3D imaging leave a lot of room for improvement. They're the wide field and confocal microscope. The problem with these methods is that only the focal plane gives high resolution information, and all the stuff in the other planes is just out of focus. In the wide field microscope, that just creates a background that gives you problems. In the confocal microscope, you use a pinhole to throw out that background, but you're still doing all of this damage, all of this bleaching in these other planes as you're going along. In my opinion, one of the most important developments in microscopy in the last decade was when Ernst Stelzer at the EMBL reintroduced the 100-year-old idea of plane illumination. In this case, instead of bringing the excitation down through the same objective you collect with, you put a second lens at 90 degrees to the detection objective, in this case a cylindrical lens, and that creates a sheet of light which you then place coincident with the focal point. Then the regions above and below are dark, and there's no damage, and there's no toxicity in those regions. There's no background from those regions. And furthermore, you illuminate the whole plane at once, so it's easy to snap that with a fast camera, then sweep to the next plane, snap that, boop, 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 and get your 3D image. That's been transformative for studying embryogenesis at single cell resolution. But it has a limitation, which is that there's a trade-off just because of diffraction between the flatness of a light sheet that you can create and how thick, over how big of an area can stay flat and how thick the light sheet is. And over the dimension of a single cell, it means it needs to be about 3 to 5 microns thick. Well, a cultured cell is maybe 5 to 10 microns thick, so you're not gaining much benefit from the plane illumination in that case. So a number of years ago, we started to play with non-diffracting beams to create these light sheets, particularly using a Bessel beam. So in a Bessel beam, you illuminate your lens not uniformly like when you're going to create a Gaussian beam, but instead with an annulus of light. And now you have two knobs to tweak, the diameter of the annulus and the thickness of the annulus. And that allows you to decouple the length of the beam from the diameter of the beam. And so now it's a pencil of light that you sweep in and out of the screen while you integrate on the camera. Then you step to the next plane and the next, and you generate your image. That sounds like it might be slow, but in our current microscopes, we go up to 1,000 planes a second or several volumes a second. So this is an example of that Bessel beam plane illumination microscope, looking at a cell which has been transfected with a cancer signaling protein that promotes this ruffling activity, which folds over the membrane and then encapsulates the extracellular fluid as these vacuoles that you see underneath in this X-ray view through the cell here. So now we're able to study these cells for hundreds of time points at fairly fast rates. There's a problem, though, with the Bessel approach, and that's, you'll never see it on this screen here, but a Bessel beam isn't just a pencil of light. In cross-section, it looks like a bullseye with these concentric rings of progressively dimmer side lobes. And as you sweep the Bessel beam across, that creates some out-of-focus excitation that you don't want. So one of the ways we dealt with that is instead of sweeping the beam continuously, we stepped it discretely, which then serially in time created a grading pattern of illumination. And once we did that, then we could apply Matz's principles of structured illumination to make lemonade out of the lemons of the side lobes and actually use the information in the side lobes to extend our resolution a bit beyond the diffraction limit, both in the sweeping direction and the perpendicular direction. So that's Bessel sim. That's what you're seeing here. One of the problems in doing that, though, is it took physically too much time to use a galvanometer to step that many times the beam across the thing. So what we did is we used the diffractive optical element to create a parallel array of 7 Bessel beams, so each one would have to go only over 1/7 of the field of view. That took care of the speed problem, but what was shocking was how much less toxic it was to use 7 beams instead of 1. And what I've learned now in doing about 8 years of live cell imaging is while the total photon dose you throw at your cell is an important metric, a far more important metric is the instantaneous power that you deliver to a cell. Seen in that light, the conventional confocal microscope, which is sort of the gold standard of live cell imaging, is probably the stupidest possible way to do live cell imaging. Because not only do you have these cones of light above and below the focus, which is doing all of this damage and bleaching, at the focus itself you have an actinic spot of light that's going, and it's leaving death and destruction in its wake as it goes along. And so the moral of the story is a line is better than a point and a plane is better than a line. So it says, why should we stop at 7 beams? So the problem is that if you have more beams and pack them in closer and closer together, you start to have interference effects between the side lobes of adjacent beams. So you have to model that. And so I started to model that, and what you find is as the beams get closer and closer together, you go through these crazy resonances and anti-resonances of constructive and destructive interference. But there are certain magic periods of the Bessel beam pattern where you get near perfect destructive interference of the side lobes. You get near perfect 100% modulation of intensity within the plane, which is ideal for structured illumination, and you spread the energy out across the entire plane. So it's really a triple win. I'm an engineer at heart, and when you get a triple win, it's like finding a unicorn. It's goddamn rare, and when you find it, you embrace it. And so this is what we do now, and this is what we call the lattice light sheet microscope. So this is some examples of using that for fast 3D cellular imaging. In the first case, we're looking at a field of dividing cells. And we're looking at it in 3 different colors at the same time. One to look at chromosomes, one to look at mitochondria, and one to look at the endoplasmic reticulum. What we're doing here is we're taking that 4D data set and we're slicing it in Z slabs about 2 microns thick each so we can see what's happening on the inside as the cells divide. Here's one such section. And you can see what happens as the endoplasmic reticulum folds up into these little pockets. The mitochondria, which are normally long sausages, fragment into pieces, and those pieces fit right inside of those pockets and they're carried along as the cell divides. So this represents 300 time points in each of 3 colors, so 900 3D volumes. Each volume consists of hundreds of planes, so it represents over 250,000 images taken from a single set of cells. And if you're a biologist, you'll know that when cells are dividing, they're incredibly light sensitive. They don't want to form replication errors. And so they have all sorts of mitotic checkpoints to shut down if they're under stress. And so the fact that these cells can take that much imaging and still divide and still be happy is orders of magnitude better than any other live cell imaging microscope. Here's another example of looking at a slime mold amoeboid, which is a model organism called Dictystelia. And these are very light sensitive. What you can see when the movie wraps around is that there's a strange little lightning bolt. When we first saw that with our collaborators, they go, what the fuck was that? You know, why are you asking us? We're not biologists, I don't know. But that's one of the things that we've seen with this microscope again and again and again with our collaborators is they are seeing the cell as they've never seen it before in 4 dimensions like this. And there are surprises everywhere. You know, you just don't know what you're going to see. We've had 50 different groups use this microscope. And everybody goes away happy and really surprised at what they see, and with 10 terabytes of data that they have no idea what to do with. And so, you know, having better bioinformatics tools, better image processing tools, all of that is absolutely essential to really harness the power of this particular technology. Here's yet another example, tetramine. It's a protozoan like you'll find in the pond. In this case, it's trapped inside of a pocket of agarose, and he's sort of frantically trying to get free for over 1,000 time points, and again, no damage to that specimen, no bleaching to that specimen. If you like these movies, this paper came out last year in Science. You can also find on Vimeo an album that has about 40 examples of this kind of thing. Or if you just Google lattice light Vimeo, you'll find it right away. So the next level up is to look at cell-cell interactions instead of single cells. So here's our good friend, the T cell we saw in turf sim before, but now not against a cover slip, but against a real dendritic cell. And so as the immunological synapse forms, eventually before there's the cytotoxic granule release to kill the target cell, there's a opening up of the actin like a lamprey or something there in order to be able to deliver it. We also find this extremely fast flow of actin away and along the sides of the T cell. Now in this case what you're looking at is 3D motility. Much of what we know about cell motility is looking at cells crawling on cover slips. But if you want to understand cancer metastasis, for example, that's a pretty poor model. So this is looking now in a collagen mesh at a neutrophilic cell then moving in 3D. And so with this, we can finally see the cells in the sort of form they really are as they try to move through tumors. And then at a larger scale, we can look at embryos. If they're small embryos, we can look at whole embryos. So this is C. elegans, the nematode worm, which is another model organism. In the few cell stage, you can see as we track the first few divisions, this particular green protein and how it localizes to the cytokinetic area with a little remnant, but also to the microtubules and so forth. In this case, we're looking at dorsal closures. So this is an entire 500 micron big Drosophila embryo, fruit fly embryo. And although we can't look through the whole embryo for reasons I'll get to in a few minutes, we can look through the top layers, and so we can actually look at both the top and bottom layer of that top layer of cells called the amnia serosa, and they go through these pulsating contractions and expansions of the actin cytoskeleton. We've been able to show that those contraction expansion phases are 180 degrees out of phase between the bottom and top surface of those cells. So the one final application to try to really push the resolution of the lattice light sheet was to combine it with our new photoactivated form of the nonlinear SIM. And so we create an activation light sheet that's structured, and then we create a readout one that has exactly the same structure, in this case a hexagonal lattice. And so by that we can extend the resolution and frequency space you can see here because of the nonlinearity. And so we've used that to do live imaging of actin, of the Golgi apparatus, and of mitochondria. But really this technique is very difficult to do because it takes 20 to 30 seconds minimum to get an image versus sub-second in the two-dimensional case. And of course you're throwing much more light at the specimen in order to work your way through the whole thing. So whether this has any real legs for biology, I'm not sure. We did it primarily as just a demonstration. Oh, how did that happen? All right, let me go back and stir it up. We're almost done. So the final topic. So the problem is going forward is that too much of our understanding of cell biology comes from looking at cells on cover slips. But they certainly didn't evolve there. So we need the ability to look at them in the whole organism where there's all sorts of cell-cell signaling and other stuff going on. The challenge with doing that is that if we're going to use, for example, the lattice light sheet, is that as the light sheet goes in, the refractive inhomogeneities of the sample scramble the light. So initially you have good resolution and good signal, but then it gets worse and worse as you go in. The same phenomenon happens on detection, that the light from these nuclei that are closest to the objective doesn't see. But the ones that are deeper down, because their light rays have to go through the specimen, they get scrambled, and then you have low signal and low resolution. So it's exactly the same phenomenon as water on your windshield scrambling the light that comes from the cars in front of you. The good news is this is a problem astronomers had to solve 50 years ago because the atmosphere does exactly the same thing to the light coming from distant astronomical objects. So after it comes through the atmosphere, you have a distorted wavefront. What they do is what's known as adaptive optics. You bounce that light off a mirror that can change its shape. You pick off a little bit of the light and put it through a sensor to measure that distorted shape. And then you close a loop in a computer and then change the shape of the mirror to exactly balance out these distortions. Get a flat wavefront, and then that gives you a good focus. So that you go from this sort of image of a star to this sort of image. And because all of your electric fields are now combining at one point, your signal goes back up to what you would get at the diffraction limit versus the far lower signal if all those wave vectors are spread across a region of space. And in the current best telescopes on Earth, because they have bigger pupils than a space telescope like Hubble, you get much better resolution than you can even with the Hubble telescope. So in doing that in astronomy, because the galaxy that's a billion light years away is pretty damn dim and doesn't give enough signal for the sensor, what you do is you shine a laser into the stratosphere to excite sodium atoms to create an artificial guide star next to that dim galaxy, and then the much brighter light from that is what goes into your sensor for measuring the wavefront distortion. A couple years ago, a group in Spain used a 2-photon microscope, which creates just a spot of light instead of a cone of light, which they could then use to create a virtual guide star. We've adopted that principle and added some other things to make this more robust, and that works very well in transparent organisms to be able to recover diffraction limited resolution. So here you're seeing a dense set of neurons in this case, first with the AO off, and you can see the low signal and then the recovery of the signal. And there's also a major recovery of resolution as you go through. So one of the challenges, though, of doing adaptive optics in biology, the challenge in astronomy is the atmosphere changes so quick that they need a new AO correction every millisecond. Fortunately, biological specimens don't change anywhere near that fast, but there is the problem that if we want to look over a reasonably large size volume, in this case a reasonable fraction of the zebrafish brain, we need to do many, many different corrections because you're going through different material in different areas. So in this case, it's 20,000 corrections. But if we turn the adaptive optics off, that's what you would see with a state-of-the-art 2-photon microscope. But now with the adaptive optics, we're able to recover diffraction-limited resolution. It's not super resolution, but it's just getting back to where we would be if these features were spread out on a cover slip instead of buried deeply inside of a whole organism. So this is an example then of looking at the dynamics of this. In this case, looking at these oligodendrocyte cells, which are sort of the glial cells of zebrafish, as they evolve, as the zebrafish is developing from 3 days post-fertilization. So we can also do that and use the 2-photon as just our guide star, not as our imaging means, and then use confocal microscopy for the imaging to do multicolor imaging. So this is looking, what you're looking as a sweep of first that fuzz was the mitochondria in a cell that's 150 microns deep, but by the time we apply the adaptive optics and the deconvolution, again, now we can look at cells deeply just as if they were cells on a cover slip, but now in the whole organs. This is doing it now, looking at oligodendrocytes and coming up the neural nuclei through the optic tectum region of the zebrafish. All right. Now, one of the limitations of this approach, just the same limitation that you have in astronomy, is you have to be able to see that guide star in order to be able to correct. Now, that's fine because the atmosphere is transparent and a zebrafish is transparent. But one of the main things people study at Janelia is mice and doing neuroscience in mice. And the mice brain, to me, as a physicist, when I look at it on a microscope, looks like tofu. And it's really difficult to get any kind of image through that. So that's why people use 2-photon microscopes, because they have infrared light that can penetrate more deeply. So what we do in that case is that we either use an infrared dye or co-transfect with an infrared fluorescent protein, and then we can use an infrared guide star, which isn't as scattered, still be able to see that at great depth in the mouse brain, all the way through the mouse cortex, and then still get spine resolution with the adaptive optics. And then a great improvement, particularly when you're doing functional imaging of neural activity by these genetically encoded calcium indicators. This is something that my wife, who's also a pioneer in this adaptive optics, uses in her work as a neuroscientist to be able to understand how the inputs from the thalamus are computed by neural circuits in the deeper levels of the mouse cortex to generate directional sensitivity to threats from different directions that the mouse sees. So that's pretty much all I have to say. One of the things that's important for me with these tools is I'm not interested in publishing papers. I'm interested in seeing my biological collaborators publish papers that answer biological questions. In fact, if I didn't have postdocs to worry about, I wouldn't publish papers at all. I would just have some kind of blog or something, or running thing of what the hell I'm doing. But those guys need jobs, so we do have to publish papers. But what we've done to try to get these tools in the hands of biologists was, first off, we've created an advanced imaging center where people can come, and we have clones of all of these instruments that I've described for biologists to use. And they come for usually a week or two at a time. They go away always with lots of data. The second is that for people who want to build these microscopes, we have complete documentation, everything down to the screws, the mechanical drawings, the wire diagrams, the control code, the analysis code, and so forth. You execute a research license and we give you what you need to build one yourself. But you need to be kind of physicist level, experimentalist savvy to put one of these things together. And then finally, commercialization. So Palm has been commercialized for ages. While there are many commercial implementations of SIM, I don't think any of them are as good as what SIM should be. And so we're trying to make sure that people have access to being able to build the SIMs. And it's really quite cheap and fairly easy to build a SIM. And the lattice light sheet, you can now buy clones of what we have from a small company that has a sub-license called 3i. And the primary licensee, Zeiss, is working on a turnkey instrument that they hope to bring out to market in a couple of years. And it's not that I give a crap about any of the money or anything like that, but I've been in business myself long enough to understand that the kind of instrument a physicist can make is not the kind of instrument a biologist wants to use. There's a lot of heavy lifting on the developmental side to make a turnkey box so he can focus on his biology and not on the instrumentation. And so with that, I'd like to close and thank particularly my postdocs. My group has averaged 3 people over the years and has never been bigger than this, which was 5. They work really, really hard, and we all work together as a team on all of the projects. So all of the work you've seen here is done by an average of 3 people over about 6 years of time. So, but it's also thanks to the contributions of all of our collaborators who come through. We've, like I said, had about 50 on the lattice scope and about 20 on the SIM. And that's one of the advantages of where I work at Janelia is it's real easy and designed to actually have 100 visitors at a time coming working on campus. and many other contributors from within Janelia, particularly Harold, who developed the poem with me, and I felt really bittersweet to get the Nobel without Harold along with it, because he deserved just as much as I did. So anyway, thanks for your time.
Thank you. I guess we have time for a few questions. I'm happy to ask the audience questions. During the time you think about it, there's one question to you. Is that the way it works? Is that biologist comes to you and says, if we were able to see this would be way through?
Right.
Is it that way it goes, or you know what? I have a neat instrument, and you could do this with that.
Right. I guess again, in my opinion, that many physicists who work in imaging are people who have hammers who are looking for a nail. Okay, that they're guided by what they know, and they're trying to see if there's a biologist who can use whatever tool that they may come up with. I think that's the wrong approach. I think the right approach is to interact with biologists, find out what their needs are. They're your customers in the end if you're going to be in this business. Find out what their needs are, and then start to think in your head about what pieces you'd have to pull together. Oftentimes those pieces are things you know nothing about, and that's where the interdisciplinarity comes in, right? Is that, to me, interdisciplinarity isn't bringing a bunch of people together because you want a physicist and a chemist and this and that. From the beginning, before the problem is defined, you want to have the problem defined, and then you want to bring in exactly the right people at the right time to be able, like what we did with Jennifer with the poem, you know, and bring in that expertise, of which was something we knew nothing about, right? And again, you find a missing piece of something you don't know about, and you just dig it out, and you find the right person who can help you out. And so I think it really has to be, you know, question-driven or application-driven as opposed to technology-driven on the other end.
It's pretty clear. Some questions? Come on, don't be shy.
They're all intimidated.
Yes. Yes.
You are working with Harald S.
Yes.
And so we are working in your case in collector physics. And so Harald S., if I don't make a mistake, was working on Bose-Einstein condensation of hydrogen. So
Of what?
He changed the subject, or is it the same people?
I'm sorry, can you say where you say it
He
here?
was working on condensation.
Yes, exactly. So yeah, Harold has barely missed out on 3 Nobel Prizes, okay? It's because he came up with the idea of evaporative cooling, which led to Bose-Einstein condensation. He published that in '85. When he was at Bell Labs, he developed an exquisite low-temperature scanning tunneling microscope with which he was able to observe these unexpected core states at the center of the Abrikosov flux lattice. And Abrikosov got the Nobel 2 years after that. And then he worked with me on the development of PALM and was left out of the Nobel Prize for the PALM. And so Harold is the smartest physicist I've ever known, the greatest experimentalist I've ever known, and one of the most modest and nicest guys you'll ever meet. There are many people who would feel bitter from that trajectory. There's not a bitter molecule in Harold's blood. So he's just happy to have contributed to all of those things. And I didn't show it, but the work that he's doing at Janelia now, he's doing a lot of electron microscopy for very large volume neural reconstructions, is orders of magnitude better than anything that anybody else is doing. And it's going to transform our understanding of neural circuits in the brain. And I've been fortunate enough that just in the last year, I've been able to rip him away from that and work with me again, because we haven't worked since really the Palm days. And now we got a crazy project, again, cryogenically, to do some cryogenic fluorescence that we're going to start working on. So Harold's amazing. I mean, He's been my best friend for 30 years, but he's a terrific scientist. Yeah. Yes.
There should be questions from data processors.
Ah.
Because
Image processing is a big
mentioned-
deal. Yeah.
you mentioned
We're
electron
swimming in
microscopy,
Right, yes.
and when I speak with an electron microscopist, I said, you really believe this is the real image? No, it's after data processing.
Exactly.
And it seems that what we've seen here is without data processing.
Minimal processing, I'd say. I mean, the SIM obviously, SIM and localization, every super resolution method involves some processing in terms of getting the result. Not as sophisticated as what you do in the low SNR regime and say cryo-EM and the particle averaging and all the rest that goes on. Although, you know, that's a testament for how great image processing can be, because cryo-EM is in a freaking revolution right now because of the cameras and coupled with the image processing capabilities. So, but really what's needed is ways of handling the big data that we have. I mean, it's fine to look at these lattice light sheet movies, but you're just seeing a surface rendering of there's all this information through the entire bulk of that thing that's going on, and we're just making qualitative insights from stuff that could be quantified out the wazoo. But we have 300 terabytes on our own personal server from that microscope alone. And most of that data just sits there, right? And there's a wealth of information in that data that's waiting to be mined, but we have no way of mining it. So, yeah. Yeah.
I read in 30 years ago, I had a technical question about the calculation of uncertainty estimation of Microsoft PAL. Perhaps we can talk about that.
Okay. Uh-huh. Sure.
I'm very interested in super resolution 3D imaging using mathematical method.
Mm-hmm.
Can you say something about that? So we have images, we have data.
Right.
So what we do is just to modelize, to find a good model.
Mm-hmm.
From a model,
Mm-hmm.
try to estimate parameters.
Right.
Finally.
Right. And so I'm not quite sure what you're getting at, but I guess what I would relate it to is, like you say, if you compare the image to a model, the more confidence you have in the model, the more you can extract from any image. So prior knowledge is the easiest and strongest way to improve your images. And if you have that prior knowledge, great. The problem is that there's not that many cases where you really have good prior knowledge. And so that's where it falls apart. I mean, a good example, again, related to the EM processing, and one of the greatest papers I've seen from localization microscopy was in 2013 when Jan Ellenberg's group at EMBL looked at nuclear pores. And remember I said the labeling density is a huge problem. And so you get all of these crappy images of the stereotypic nuclear pore with 7 or 9 subunits around it and only some of them labeled on this one and that one. But you look at 5,000 of them and you know a priori that they're a stereotypical structure. They're all going to be the same. Then you can do the same particle averaging techniques they use in cryo-EM, and then you get to average out all of that shitty labeling, and then you're able to determine the position, in their case, of about a dozen different proteins, their radial location within the nuclear pore to almost an angstrom precision. And by doing that, they were able to determine a subunit inside of the nuclear pore in its orientation that was ambiguous from the cryo-EM data. But by fluorescence microscopy, they were able to do something cryo-EM was not. But it was, again, with the prior knowledge that they had a stereotypic structure to work with.